Hello everyone, my name is Osama, and today I'm going to talk about the Krebs, which is also known as the TCA cycle and the citric acid cycle. Now, let's start from glycolysis. When glycolysis ends, where pyruvate is produced, and it's present in the cytoplasm, since glycolysis occurs in the cytoplasm. Okay, so we need to transport this pyruvate into the mitochondria. How do we transport it? We have voltage gated pooling complexes in the outer membrane, and then it passes through the mitochondrial pyruvate carrier in the inner membrane, and this is how the pyruvate ends up in the mitochondria. Now, why do we want it in the mitochondria? Because of the link reaction. The link reaction is basically the reaction that links glycolysis to the Krebs cycle, and that's the link reaction right here. So it converts pyruvate into acetyl-CoA, and it produces CO2 and NADH2. Just NADH, not NADH2. This reaction, of course, occurs in the mitochondrial matrix. Like we said, we transport the pyruvate into the mitochondria. It involves pyruvate dehydrogenase complex, which is also known as PDC or PDH. Now, this complex contains three coenzymes. The E1, which is a decarboxylase enzyme. This is the enzyme that removes the CO2 from pyruvate. The E2 enzyme, which is the transacetylase enzyme. It transfers the acetyl group to coenzyme A. And E3 is a dehydrogenase enzyme, and it reduces NAD by adding the hydrogen to produce NADH. Now, PDH requires, or pyruvate dehydrogenase requires 5 coenzymes, or also known as cofactors, and use this mnemonic to memorize the names of all 5 coenzymes, the lovely coenzymes for nerds. These are the coenzymes. We're gonna talk about them in more detail in the next few slides. Okay, the first coenzyme is thiamine pyrophosphate. It's derived from thiamine, or also known as vitamin B1. It's a cofactor for 4 specific enzymes. Now, for today, we're gonna focus more on these 2 enzymes, but these are other enzymes that thiamine pyrophosphate can act as a coenzyme for. But today, we're gonna focus on pyruvate dehydrogenase, which is in the link reaction, and alpha ketoglutarate dehydrogenase, which is in the TCA cycle. Okay. And now, a deficiency in thiamine or vitamin B1 will cause decreased production of ATP, of course, due to decreased rate of link reaction and decreased rate of this step in the TCA cycle, which will cause 2 diseases. The first one is beriberi. Beriberi is more common in underdeveloped areas, and there are 2 types. Dry beriberi, which causes polyneuritis, which is inflammation of multiple peripheral nerves in the body, and muscle weakness. Wet beriberi causes tachycardia, high output heart failure, where the heart has a high output of blood, but cannot accommodate the body's needs, and edema, which is fluid buildup, especially in the lower limbs. Now, Wernicke-Korsakoff syndrome is more common in alcoholics. It causes 2 distinct symptoms, which are confusion and confabulation. Confabulation is a symptom where the patient creates stories that he believes in, but the stories aren't true. Now, to treat thiamine or vitamin B1 deficiency, first we have to replenish the thiamine stores, because even if we give them more glucose, they can't oxidize it. So we have to give them thiamine first, and then we give them glucose, because if we just give them glucose, it will increase the metabolic demand, but the body cannot keep up because it doesn't have thiamine or vitamin B1, and the enzymes won't work. Now, this is the second coenzyme we're going to talk about, which is FAD. It's derived from riboflavin or vitamin B2. Now, riboflavin is added to adenosine, and that produces FAD. FAD can accept electrons to become FADH2, and it is a nucleotide enzyme. Now, NAD plus is also a nucleotide enzyme. It's derived from niacin, which is also known as vitamin B3, and niacin is derived from tryptophan, the amino acid. Okay, it accepts same as FAD. It accepts electrons and hydrogen to become NADH. Now FAD and NAD, they both FADH2 and NADH2, they both can be used in the electron transport chain. Now, a deficiency of vitamin B3 will cause a disease called pellagra. This disease is characterized by the three Ds, the three symptoms: dermatitis, diarrhea, and dementia. Now, the fourth enzyme is coenzyme A. It's derived from pantothenic acid, which is also known as vitamin B5. It accepts or donates acyl groups. In this case, it's an acetyl group, and it is also, just like FAD and NAD+, it's also a nucleotide enzyme. Now, these coenzymes, they're all derived from B vitamins, but something important to know about B vitamins is that they're all water soluble, and since they're water soluble, they can be easily washed out and excreted in urine, with the exception of B12, since it can be stored in the liver. So, since they're continuously excreted in the urine, they have to be continuously replenished too from the diet. So you have to consistently supplement with B vitamins. That's why beriberi is more common in underdeveloped areas where there's less nutrition. Now the fifth coenzyme, this is the only enzyme that's not derived from a vitamin. It's lipoic acid. It's a cofactor for E2, and it can be inhibited by the poisonous metal arsenic. This metal binds to lipoic acid and it inhibits PDH, the enzyme complex. Okay, this will produce a very characteristic symptom, which is garlic breath, due to arsenic being oxidized to arsenous oxide. Now, how do we regulate PDH? We basically regulate PDH to regulate the link reaction. So when the body is in a low energy state, which can be signaled by high ADP or high NAD plus, or in a high energy demand, for example, high calcium ions in muscles, PDH is activated. But when the body is in a high energy state, we don't need energy, we have high ATP, it's signaled by high ATP, high NADH, and high acetyl-CoA. Or in a high fatty acid oxidation state, PDH is inhibited. Why do we inhibit PDH in high fatty acid oxidation state? Because if we have enough energy from fatty acids, why would you need to oxidize the glucose? So that's why PDH is inhibited. Now, how is it inhibited or activated? PDH kinase is an enzyme that phosphorylates PDH, and that inactivates it. And PDH phosphatase will dephosphorylate PDH, and that will activate it. So PDH is active when it's dephosphorylated, and it's inactive when it is phosphorylated. Now let's talk about the Krebs cycle. The Krebs cycle is an amphibolic cycle, and what that means is that it's involved in both catabolism and anabolism. Now in catabolism, it oxidizes acetyl-CoA, which is produced from the link reaction, into 2 CO2, 3 NADH, 1 FADH2, and 1 GTP. Now this is per turn. It may have multiple turns for a single glucose molecules. And for anabolism, the intermediates of the TCA cycle can be used in biosynthetic synthetic reactions. And these are examples. Of course, you have to know them. Now let's talk about the first step. We'll talk about a few steps in the TCA cycle, not all of them, but we're going to talk about the most important steps. Now the first step we're going to talk about is is catalyzed by the isocitrate dehydrogenase enzyme. This is a step, rate limiting step, so that means it's highly regulated. And this is a oxidative decarboxylation reaction, which means that it's a redox reaction, and it's also a decarboxylation reaction since we produce CO2. It's a redox reaction because we produce NADH, and it's also the first NADH producing reaction. Now, since it's a rate limiting step, it's highly regulated, it's activated by low energy, it's signaled by high ADP, or muscle activity signaled by high calcium ions. But it's inhibited by high energy, which can be signaled by high ATP and high NADH. It requires the coenzyme NAD+, the same one that's required from PDH, and it's also derived from vitamin B3 or niacin. And like we said, the deficiency causes a disease called pellagra. Now let's talk about alpha ketoglutarate dehydrogenase. This is also a rate limiting step, and it's an NADH generating step. Like as you can see here, it generates NADH plus. I mean NADH. It's regulated the same way isocitrate dehydrogenase is regulated, since it's also a rate limiting step. It requires the same cofactors as PDH. Now, you see these, the same coenzymes that were required by PDH, they're also required by this enzyme. And it can be subject by to feedback inhibition. Like for example, if we have enough succinyl-CoA, that will inhibit this enzyme, so the rate of this reaction is lower when we have enough succinyl-CoA. So that's what feedback inhibition means. Now for the next enzyme, it's succinyl-CoA synthetase. This is the only substrate level phosphorylation in the Krebs cycle, and it produces GTP. We phosphorylate GTP, we add an inorganic phosphate group to produce GTP, guanosine triphosphate. And for the fourth enzyme, it's succinate dehydrogenase. It's the only enzyme that's not located in the matrix. It's located in the inner mitochondrial membrane, and it's also part of the complex 2 in the electron transport chain. This reaction is the only reaction that produces FADH2, and it requires of course, since it produces FADH2, it also requires the coenzyme FAD. And in the case of riboflavin or vitamin B deficiency, the symptoms they're going to be caused by it are glossitis, which is inflammation of the tongue, stomatitis, inflammation of the mouth and lips, and chelosis, which are cracked lips. And since this enzyme is in the inner mitochondrial membrane, it directly links the TCA cycle to the electron transport chain. Since the electron transport chain is in the inner mitochondrial membrane, FADH2 doesn't have to, it just stays where it is. So it doesn't have to move anywhere. Now, in the Krebs cycle, we have anapleretic reactions. These reactions, what they basically do is they replenish the intermediates that are being used for the biosynthesis reactions. So, for example, when oxaloacetate is used in biosynthetic reactions, we're going to have less of it. But how do we keep the cycle moving? We convert pyruvate into oxaloacetate, and the enzyme that does that is pyruvate carboxylase. Now this reaction occurs in the mitochondrial matrix, and it's biotin dependent, and this reaction is activated by high acetyl-CoA. Now if you look here to the Krebs cycle, if we have high acetyl-CoA, that means the cycle isn't moving, which means we need more oxaloacetate. So this reaction helps us produce more oxaloacetate so we can keep the cycle moving, and if it's not moving, it will be signaled by high acetyl-CoA. And thank you everyone. Good luck with your studies. Thank you. Bye.